So in lab 5, you are going to work with two enzymes. So the first one we've talked the most about was catalase, right, with the potato. The second we're actually looking at is called amylase. Now amylase is an enzyme that specifically breaks down complex carbohydrates. So you have a specific type of amylase in your saliva, in your mouth, to help break down starches before it hits the rest of your digestive system. So this reaction simply is that starch, a complex polysaccharide, complex carb, gets broken down into reducing sugars. So we take the pieces of starch off of themselves. So like glucose is bound together to form starch. We're going to break those glucoses up to give us reducing sugars. This is a hydrolysis reaction because we are breaking, right? Water will come off and we will have simple sugars. Now there's an enzyme that's amylase that helps to speed up this reaction. So amylase is an enzyme that causes starch to break down into reducing sugars a little bit faster. So in order to do this test, it's not as simple as the catalase. You can't just watch bubbles go, but you do get to look at pretty colors. You are going to have to run two tests on this. So you're going to have to run one test that is about starch and one test that's about reducing sugars. So if you remember back to that lab that we did with our biological molecules, we have two different tests, right? We have an iodine test, which is going to test for starches, and we have a Benedict's test, which is going to test for reducing sugars. So because of that, we have to make two different test tubes containing the exact same thing. All right, so if you look at what happens to our test tubes here, all right, test tubes 1 and 2 are exactly the same. One is going to get tested with iodine, the other with Benedict's, to see if there's starch and to see if there's sugar. Same with test tubes 3 and 4. They are just starch mixed with water. We'll test one with iodine to see if there is starch, and we'll test one with Benedict's to see if there are reducing sugars. Then 5 and 6, we're going to be actually looking at the enzyme, which is amylase. So we'll mix starch and amylase, and one will test to see if there's any starch, and one to see if there's any sugars. So I've run this test. All right, this has to sit for a while, so I let mine sit for a good long time in order to let the enzyme work, because catalase worked quickly, right? I could do that in film in a couple of minutes, right? It was pretty quick. This takes some more time, right? At least 15 to 20 minutes and a little longer if you can. So what I want to do is compare test tube 1, 3, and 5. And then test tubes 2, 4, and 6, which is what we're looking at right now, right? So test tubes 1, 2, and 3. Test tube 1 was tested with iodine. It's just water, right? This is just the color of iodine, which means this is a negative test for starch. Test tube 3, remember, is just starch mixed with water, right? When that occurs, you can see that there is a ton of starch, it's basically black. And then test tube 5 was starch mixed with amylase. So remember what should happen. Starch should get broken down into reducing sugars. So starch should disappear. In this case, it did. Now, it didn't go away entirely, right? You can still see some of that in there, but most of this is the color of iodine. So there's a little bit of starch present, but not much, which means starch was able to be broken down by the amylase in test tube 5. We went from a lot of starch, which is test tube 3, to just maybe a little bit of starch that's left in test tube 5. Right? So what we saw is that amylase was able to digest the starch and go through the hydrolysis reaction. So then let's look at our Benedict's test. Test tube 2 was blue, which is a negative test for Benedict's. All right, test tube 4, also a negative, right? So neither test tube 2 or 4 have any sugar in them, right? There's no sugar present. Now in test tube 6, though, tons of it. Look at that, how orange and brilliant that is. Basically, again, as we look at what test tube 6 should be, all right, this is starch mixed with amylase. So what's the job of amylase? Is to break it into reducing sugars. The Benedict's test tests for reducing sugars. Therefore, we have a bunch in here, right? So test tubes 1 and 2. Right, these 1 and 2 are the water, right? They were a negative control, right? This was to show us what our negative should look like. Test tubes 3 and 4 looked at what happens when starch just sits in water for a half hour or however long you let this sit. We still have starch present. There is no sugar that's going to be present there. But the big story really is what happens when you take starch and mix it with amylase. So when you mix it with amylase, the starch disappears. The starch is hydrolyzed, less of it is present, and more sugar is present. We get sugar when the amylase is allowed to work on reducing sugars, or work on the starch to produce reducing sugars. So again, we looked at duplicate test tubes to determine if hydrolysis would happen when we use amylase. And it did, right? We saw it quite clearly here. Starch can't break down without the amylase, but it can break down into reducing sugars with the amylase.